Thank you. Hi, everybody. First, off the top, I'm a social scientist. I'm not a physical scientist, so I'm learning a lot today from all of you. And I'm going to be presenting data, but there's a little danger there. So just in that context I'm going to be talking about some science that you all probably know more about than I do, but some other things that I'm doing. Real briefly, kind of an agenda. I call it a rapid agenda here, because it's going to hit on a bunch of topics from a bunch of different angles. And this is condensed from a presentation that's maybe 4 times longer. So I'm sorry in advance if I hit something too quickly, but happy to loop back either today or anytime and talk about it more. My goal today is really to share the data we've got with all of you in hopes that you can use it productively and to get some feedback, again, now or later, because we're really working on understanding where and how the data we're producing can be most valuable to people. So I'm going to touch on four aspects of what we're doing: the geography of where we're doing it, sort of the data structures, the methodology, and then actual results in the Klamath Basin, and then discuss. Let me say 2 things to begin with. Again, in that context, my academic training is at the Harvard University Kennedy School on environmental economics and environmental policy. And so a lot of the topics we talked about earlier, to me, I hear as what's the value that someone influential is getting out of nature, and how do we translate that value into behaviors that result in a more vibrant life on Earth for the planet and for the people. And a big piece of that for me actually focuses on the economics of nutrient cycles between the environment, agriculture, food, and human health. That's not really what we're talking about today, but that's what's going on in the background of me. In terms of geography, here's a quick map of the Upper Klamath Basin, and each dot on this map is one of the monitoring sites that we're managing, that we've implemented. I'll explain what that means in a few minutes, but you can see where they are. I'm going to explain why BeeOdiversity, the word bee is in our title, but it's basically the key insight here is that we use bees to gather samples from the environment. We use bees to gather billions of samples per year per site. We're going to go into that a little more. On this map, there's 13 sites that are solid dots and 6 of them that are little hexagons. Again, don't worry about the details, but let me point out that the solid dots are domesticated honeybees. The hexagons are wild bees, wild solitary bees in the landscape. And they have different habits and they cover different territories. So we'll talk about that a bit. I'm going to show you one more map because this is where we're monitoring today, this year. But we started this 3.5 years ago in a slightly bigger geographic range as we've sort of tried to pilot the approach and figure out where and how and with whom to do it and stuff. So this is a little bigger map. And the reason I want to show this to you is because we did run some pilots 3 years ago down in the Happy Camp and over in Etna. And so the data generated there is still part of the data set. And a couple of those folks I see your names on the call today. So it's good to know that you're there virtually. So what do we mean in terms of the data? Again, just a snapshot. We are producing data. Again, in a few minutes, I'm going to tell you how we do this and why and with what parameters. But basically, these data types, plant species, pesticides, metal elements, and what we call specials, which means we're experimenting with some other things we're trying to detect with scientific and statistical reliability. These are actual results from our work in the Klamath Basin. So again, without diving into too much detail, what you see down the left-hand side is the top part of a list of plant species that we've identified. We're up to about 330 distinct species between the sites I showed you on that map. And for each of those plant species, we identify each year the location and the dominance of that species in the environment based on our sample. So 330 distinct species. which you'll see in the interpreted section, we then analyze to say lots of interesting things about your ecology, threats and opportunities, wildlife habitat, etc. We've also identified the 15 pesticides listed here. We're going to talk about all this a little more. Each of these are identified in parts per million concentrations. We've identified the 7 metal elements listed, and we're experimenting with dioxins, 6-PPDD, and microcystins right now. So one more completely illegible spreadsheet, which is to say this takes the plant species list and breaks it down into a bunch more detail. Conceptually, what I want you to see is that each column on this sheet is one of those different sites. And in fact, there's 4 columns per site because we're measuring 3 or 4 times per season. So we're tracking the data, either the plants themselves or the chemical or contaminant residues that we pick up on in the pollen of those plants, which is pretty precise. We're tracking it 3 or 4 times per year per site, and then plotting the presence, the trends, the dominance, the emergence, things that are new, things that are old, etc. That's kind of what the raw data looks like. Next, the methodology, just briefly. We call it biomonitoring. And some of you, because I see Michael's name on this list, know a lot more about bees than I do. So I'm going to give you the snapshot, which is that a domesticated honeybee like this, a hive of bees like this, covers about 1,800 acres and brings in about 1 million environmental samples per day. What we do is we site a couple of beehives managed by local beekeepers who we provide a little bit of training to and a little bit of equipment. And basically, 500 million to 1 billion samples are collected in the course of a season, and we analyze that information. I'll explain more. Here's some real life pictures from the Klamath Basin, just to show you what it looks like on the ground. The top middle picture is me, and then you might recognize Carl Winter of Lakeside Farms, Lorena Corzat of the Klamath Basin Beekeeper Association. These are the two data collection stations on Lakeside Farms on the east shore of Upper Klamath Lake. These have been operating for 3.5 years now. On the right is the sample, 1 million or so samples, in fact. On the bottom row is the wild bee technology that I mentioned. Again, our purpose today isn't to go into this in detail, though I'm really happy to. But this is also a device we developed and we just piloted this year for the first time at six sites between Iron Gate Dam site and the Williamson River Delta on Nature Conservancy property in Upper Klamath Lake. And the intent here is to attract wild solitary bees to different nesting conditions, which different species of bees are attracted to record their arrival, their presence, their in their diversity and their abundance during different points of the summer using photography, and then also to be able to extract little sample tubes for digging into precise species analysis and food sources. And then we analyze that to tell you, amongst other things, not only about the bees, but about what that information says about the ecology and the ecological health of the habitat where you're working. This photograph is Dr. Kim. Dr. Kim is the founder of our organization, and he runs a lab at the University of Liège in Belgium, where he developed this process about 10 years ago. So again, happy to discuss, but on the right-hand side are the kind of data and metrics that we tend to report out. We then use a series of our online data dashboards and such to provide the data to whoever we're partnering with on the ground. I'm going to show you now 2 examples how the data kind of breaks out. These examples come from Lakeside Farms, thanks to Carl. And notice at the top is example 1 of 20. We're not looking at 20 today, we're going to look at 2. But there's so many ways to slice and dice the information that I just wanted to show you a couple. These 2 charts compare the year 2023 and 2024, and they're looking at plant families represented in the data. And you can gather what you want to gather from that. There's lots of things. In this case, on the far right, you'll see we also have the data from 2022. By working with Carl and looking at this data, Carl's been able to diversify the range of plant species on his property, not only favoring the restoration of the wetland along the river and of wild species or their native species, perhaps the elimination of certain invasives, but also recognized a real dearth in the late, what we call P3 and P4, the late periods of 2022. And in fact, the year before we also worked, that's when we started on Carl's property. You fast forward to 2024, you see that there is a much higher representation of different plant families on the property. Some of that may be due to exogenous factors, including the fact that Carl's restoration work is attracting waterfowl that are bringing in new and interesting species. Some of it is also very intentional based on this and other data. My point being, it's interesting, and this is really what my organization does, is tries to support individual influential people on the ground who are actually taking restoration action. And so that's the framework with which we've approached this. That's how we arrived at the map that I showed you at the start. But what you're going to hear in a minute is that we've also, with some coverage now, have begun to work on rolling up those individual site data points into some regional data snapshots and such. Example number 2 of 20, just to give you another example of how the data can be used, is actually assessing at Carl's property at Lakeside Farm, the amino acid content of the different plant families reflected in his data in order to determine what the nutritional profile is for pollinators across those 1,800 acres. So pollinators, in our case, are our partners in collecting samples. They can also be a beneficiary. It's far from being the only source or purpose, but in this case, the red bars on the graph basically indicate that at the end of each year, 2022, 2023, 2024, there's a bit of a risky drop in certain proteins that pollinators need. I know that one of them that we see the drop in on this site is tryptophan, and I can't remember what the other is. So the action point at Lakeside Farm is to work on cultivating some plants that carry those missing amino acids in order to bolster the nutritional capacity and carrying capacity of the landscape. There can be other uses of this information, but that's what's going on there. Finally, in terms of me breaking down the way we're organizing the data, I told you we're beginning to try to roll up this information into sort of regional views. So again, I'm going to show you some summary maps and then dig into some specific data points a little more than stop. So on the Klamath, a couple things. One is what we've done is on the map you've seen, and this comes from our 2024 analysis. By the way, we were going to wait to offer this presentation until later when we have all of our 2025 data assessed and presented and have the permission from all the landowners. But a couple of people have expressed interest, and we have an interest in trying to get more sites operating. And if so, we kind of want to start those before bee season, which is mid-April or so. And so we decided to present this to you now, even though it's not quite all complete. So we mapped land cover as a reference case at each of the sites. Therefore, you can see that the relevance of the data at the site level is somewhat contextual based on the ecosystem condition we're operating within, and they vary quite a bit. Look at the Sprague River compared to Klamath Falls town, or even Lakeside Farm on the lake, et cetera. then some of the metrics that we report based on the data and then go over with the landowner and others, if interested, are species richness, total number of plant species on the property. That's what these numbers indicate. Again, we could interpret them more. Wild species ratio, what portion of the plants on the property are considered wild versus non-wild or non-native or even noxious. So you can see it varies fairly substantially between properties. And again, this influences the stakeholder actions that people take, that we work with them on. We're able to isolate out plants that are favorable to wetland or water conditions. And so depending on your interests, and by the way, all of our sites are on watersheds. There's water running through every property that we're working on, whether it's the Klamath itself or small creeks up in Bly, Oregon, or others. So you see the number of classified wetland favored species at the different sites. We see noxious species according in this case to the Oregon state classifications. We see indicator plants of disturbed environments. We see pesticides. Just flip over to that for a minute. In this case, we have fungicides, insecticides, and herbicides. You see this is the number of pesticides per site identified. We then correlate that pesticide data, I'll stop for a minute, with a number of things, depending on, again, on your interests. We scan for acute levels of toxicity or redundancy in pesticides. Maybe multiple pesticides are being used to treat the same environmental condition. For example, we frequently find 2 or 3 different fungicides being used for the same purpose, in which case we can work quickly with the private or public site manager to pick one of those and stop using the other 2 and instantly reduce at least the number of different toxic substances on the property. We calculate the groundwater ubiquity score, groundwater leaching index for each of the pesticides identified. So in this case, H is high, M is medium. With respect to the pesticides we identified, kind of see here. Oops, there we go. You'll see up there on the Wood River, we found the presence of one pesticide at a concentration that indicates a pretty, a serious enough risk for groundwater leaching that we're doing a bunch more testing up there this year, and others who have what we classified as the medium level of risk. The NOEC, the no observed effects concentration on relative to fish, linked basically to 2 pesticides that we found in the basin, carbendazim and pendimethalin. There's 3 or 4 sites we rated as a high risk, meaning we label these as points for attention. We're not saying they are or are not a problem per se, but we're advising the site manager or our partners to take a harder look at what's going on and to consider some remedies. We've also looked at the acute toxicity index and sort of found a medium level of concern at many of the sites throughout the basin. Finally, and then these are my last couple of slides in terms of the overview. This one presents more details on the pesticides and the heavy metals we identified. Not expecting anybody to study this chart in detail, but I guess what you can imagine is 72 columns further to the right that break down each site by each time period, by each pesticide or metal element, and list the parts per million concentration identified in each of those slots. So I rolled all that data back to this table just to try to illustrate a few things. The next slide is the same, but I've flagged a couple of things to pay attention to that we grabbed out of it. One is I'm going to talk about each of the orange colored cells for a minute. The top row, DEET, right? Mosquito repellent. It's everywhere. It's everywhere. It's in the most remote site you thought you wouldn't find it in, and it's in the middle of town. So it's just quite interesting. You'll see that in the range, again, this is a very crude summary, but the mean, the median are around 0.03, but we've found it in certain time periods up to 0.15, which is a lot a lot higher. So there are some heavy concentrations in a few places that we then again work with the localized situation to try to figure out what's going on or why, or if that actually poses a risk. The couple of pesticides I mentioned earlier are highlighted on the list. On the left, I would also point out this year that so we've been doing this spot testing it and then further developing it since late 2021. New data shows up. So these two here that I can't even pronounce showed up in the fall of 2025 for the first time ever. So to us, that's on a site that we'd already been monitoring for 3 years. So new things, changes are happening. New things show up. The data is dynamic. If we go down to the metal elements for a minute, I highlighted chromium because again, you'll see that the average levels, it's present everywhere. It's on all 18 sites. The only one that's not is, so we did not find mercury anywhere. I want to say that. We always scan for mercury. We've never found any mercury in the basin. Cadmium only showed up at 17. These other 5 show up at all the sites. But look at this chromium level of 1.6 compared to these others. There's something going on that we just picked up on this year because we saw a drop in chromium on a couple of sites, and we started investigating why compared to the previous year. And what we're just processing now is that the whole Upper Klamath Basin saw a spike in the whole basin saw a pretty big spike in chromium in the summer of 2024, and then it dropped again this year. I have no explanation. I have no more details at this point, but it's pretty interesting to observe. And then lead is highlighted down below, again, because at a couple of specific sites, the level's pretty high. It's below the EPA regulatory limits for lead content in soil. However, it's at a level that we really only see in industrial sites in urban environments usually. That's just at a couple of places. Things we're also working on this year are dioxins, 6-ppd, and microcystins. We've documented in the laboratory that we can detect all of these. The question is, are we going to find it? Can we find it? And at what levels of limits of quantification in the laboratory? This is my last slide. Love to hear from you now or later. Which of this stands out to you as being useful? I really need to hear that. Do you see gaps in the geography? What other information could we correlate it with, or any other questions or comments you have? Thank you.
All right. Thanks, David. Super interesting. I'm going to start off by asking the question about microcystin. This is a cyanotoxin found in water. So how is that connected to bee pollen?
Yeah, so, sorry, I'm gonna, here. The question here, so our testing is super sensitive to airborne emissions of lots of things. And so the question is, the question we are posed with is, can we help identify locations, concentrations, and drift of microcystins by looking at what is settled on the pollen? So, for example, I mentioned the chromium throughout the basin. That chromium had to be in the air. It's sites that are 70 miles apart from each other. There was some incident of some kind somewhere. We've also, in a 5,000-acre organic ranch in Montana, we found what turned out to be lead dust from a mine in the hills 22 miles away that only blows in the springtime when the prevailing winds come from that direction. things like that. It's pretty interesting, the sensitivity of this analysis to those airborne particulates. Therefore, can we do something useful with microcystins? We're not sure. But we heard the question often enough that we're like, well, let's try it. Let's see what we figure out. And we actually talked to Fish and Wildlife last week, and they pointed out that our sites where we're doing the testing are probably not optimally located to identify microcystins. So we're having a conversation now about, okay, good, because that's not our specialty, right?
Yeah.
Yeah.
And Chauncey has a question in the chat. Were those metals and pesticide concentrations in the tissues or in another medium, like soils or water?
Yeah, so they're in the pollen.
Yeah.
In the pollen that the bees are bringing back. Yeah. So depending on which one we're looking at, we know that certain metals can come through the soil all the way to the pollen, and will metabolize all the way to the pollen. Others won't. So we can do some investigative work to hypothesize that the source of this was soil versus air or water. But at some point we stop and have to turn to local intelligence. And we also do soil tests, right? I mean, we'll actually do soil microbiology and contaminant tests now to correlate with this data. Not here in the Klamath, actually in places like South Africa and a couple other places where we're doing this work.
Okay, great. Any other question? Oh, here's one from Maya in the chat. Have you found that wildfires could be sources of metals, including chromium, in other watersheds?
So this is our first work in the Western United States. I haven't given that whole history, but this was originated in Europe. We brought it to the US starting 3.5 years ago. So it's been a learning experiment. We now have about 50 sites around the country. This accounts for 19 of those, right? So a little context is we haven't worked in a lot of wildfire country. I will say, however, that in Klamath, 3 years ago when the smoke was real thick, we found some stuff that people think must have come in on the smoke because there didn't appear to be other sources and it's gone away. Let me mention that in that same year, we also found a residue of something that took about a year to track down, and it turns out to be it's a residue from smokeless gunpowder, and there's a skeet range about a mile away. So we're hypothesizing that gunpowder residue from the skeet range is making its way to the pollen on the plants that the bees are bringing back to the hives.
Wow, fascinating.
Yeah.
Another question in the chat. You mentioned an EPA criterion. Was that specific for pollen?
No, thank you. We use depending. The USDA has some criteria, although those vary by state sometimes. EPA has health consumption, food and water. So we're able to report against whatever standardized criteria we can identify. It's a reference point. It's a point of information. It's not right. Yeah.
Yeah. And building on that, when you talked about correlating soil samples to the bee pollen samples, do you see higher concentrations in the pollen than you do in the soil? Is it getting magnified?
Well, there's two things we're doing. We're correlating the actual result, and then we're correlating the biodiversity of the soil with certain outcomes. So, for example, the case I mentioned in South Africa, we're actually correlating water retention in an arid environment in the soil with the biology of that soil, and then looking at how basically stands of invasive trees have changed the biology of the soil. So whereas the landowner, which is a 5,000 acre, 8,000 acre commercial agricultural product company, thought that the trees were sucking up all the water and that was the problem, we think that the real problem was the trees changed the biology of the soil. And that's the problem, right? So just, sorry, I didn't actually answer your question, but it's a pretty interesting result. Yeah, I don't know the answer. I'll have to ask Dr. Kim about your specific question. Let me mention one more thing. I wanted to say this because something was mentioned earlier. which is the way we do this work using the bees to gather very objective data. As a social scientist, is a pretty amazing stakeholder convening mechanism. Lots of people gather around the table who might not often talk to each other when we bring this data set to the table. It's really interesting and fun because you get really vibrant, honest engagement based on, this data from the bees on your property. So it's a pretty powerful tool for organizing as well.
Great. Well, David, I want to thank you. It's a very interesting and unique talk. So good luck with the rest of your project here.
Thanks, and thanks for teaching me a lot today too.